my name is Buyanzaya Buyanurt. I'm a rising fourth year pursuing a bachelor's in environmental studies and a minor in chemistry. I'm a Gorman research scholar, currently working in Dr. Gretchen Hoffman's lab under the mentorship of Terrence Leach. This summer, we wanted to examine the effects of varying pH conditions on the early development of the painted sea urchin, Lidocainus pictus. The consequences of global climate change threaten the survival of many marine organisms. Climate change due to greenhouse gas emissions contribute greatly to ocean acidification and ocean temperature rise. Ocean acidification is basically when the water becomes more acidic. These events, in turn, impact calcifying organisms such as sea urchins. Calcifying organisms use a chemical compound to build their skeletal structures. And basically, when the water is acidic, these chemical compounds are limited in abundance. So we hypothesized that calcifying organisms So we hypothesized that these organisms are more vulnerable to other environmental stressors and acidic conditions. So by the end of the summer, we wanted to answer the question of whether early exposure to varying pH conditions had altered the tolerance of our Lidocainus pictus larvae to an acute heat stress event. My goals for the summer were to successfully raise viable sea urchin offspring, manipulate multiple variables such as temperature and acidity in my experiment, and also lay down the framework for further research on Lidocainus pictus. So why did I choose Lidocainus pictus as my model organism? It's not because they're cute. So there's three important reasons. First, they're an ecologically relevant species. Second, they're a good developmental model. And third, they make babies or spawn in the summer, which is convenient. So my project required me to first spawn my urchins, then raise them in buckets with varying Pco2 levels, and finally introduce a heat stress event. So the spawning procedure begins with injecting potassium chloride into our adult sea urchins. This potassium chloride solution basically causes the urchins to contract their muscles and excrete either their sperm or eggs. And we can determine the sex of our urchins by looking at the color of their excretions. White for sperm, orange for eggs. So we then sample these eggs and sperm and pull them together into one beaker. We give them time to fertilize, and under a microscope, we check for a successful fertilization. And we can tell that it's a successful fertilization if we see this nice circular fertilization envelope around the eggs. And lastly, we want to make sure that at least 80% of our fertilized eggs in the beaker are successful. And then we move on to the next step. So we take our fertilized eggs and we divide them up into 6 buckets. 3 buckets with high Pco2 conditions and 3 buckets under low Pco2 conditions. So I'm going to walk you guys through Pco2 and pH. This can get a little confusing for some people. So Pco2, or the partial pressure of carbon dioxide, basically measures, how much carbon dioxide is in the water, the concentration. So with a high Pco2, we're going to have a low pH. That means the water is more acidic. So although our pH numbers are only 0.3 units apart, we know that these two conditions are drastically different from one another by looking at our Pco2 numbers. And we chose these conditions. Low Pco2 is ambient conditions today, so present day water conditions. And our high Pco2 is predicted water conditions in the future. So when our embryos have reached their larval stage, we subject them to a heat stress event using this thermal block right here. We divide up our larvae into 8 vials, 1 row for each Pco2 condition. And this thermal block kind of works as a gradient from cooler temperatures to hotter temperatures. And we let them kind of sit in these hot, in the stressful conditions for an hour. And after this hour, we look at each individual vial under a microscope. And we give them, and we determine whether they're alive or dead by looking at their body movements, or normal and abnormal by looking at whether they're pretty or ugly. So this experimental process allows us to measure 3 parameters. Morphology, which is basically measuring their skeletal length, the length of their skeletal structure, thermal tolerance, and also their developmental success, whether they're forming a nice gut and forming nice skeletal structures. So here's a little sneak peek at our results. So to get you guys oriented with this graph, on this y-axis we have our proportion alive or normal, and on the x-axis we have temperature. So each of these points represent an individual vial in our thermal block. And if you guys remember from what I said in the previous slides, we gave them, we scored them by alive or dead, normal or abnormal. And we fit a linear model to our data, and from that we were able to measure our LT50. So LT50, or the lethal thermal limit, is basically when 50% of our population has died. And our individuals that developed under high Pco2 conditions reached their LT50 at 31.6 degrees Celsius. And then when we compare it to our individuals that developed in low Pco2 conditions, we can see that their LT50 was at a lower temperature of 31.1 degrees Celsius. So we can kind of see from here that our high Pco2 individuals are actually doing better. And we also decided to measure abnormality. So AT50 is when 50% of our population looked abnormal. And we found that our high Pco2 individuals reached that limit at 29.6 degrees Celsius. And then when we compare it to our low Pco2 individuals, we see that they reach it at 29.1 degrees Celsius. As we can see from our data, individuals that developed in high Pco2 conditions had a much higher tolerance to thermal heat stress. So, will we see these higher ocean temperatures within our lifetime? Probably not. So, is my tolerance data relevant? Yes, it is. So, as you can see here, the average summer temperatures from 2014 to 2016. They fall within a range of 16 to 20 degrees Celsius. And we can see that here, in these temperatures, our Lidocainus pictus larvae are resilient. They're doing well. But we also see these max summer temperatures that go up to 23 degrees Celsius. And here we see that it falls within the range of where abnormal development can occur. So why is abnormality important? As some of you may recall from our previous slides, here's a normal larvae and here's an abnormal larvae. So a normal larvae looks really cute and it has a nice spherical gut. And here's our abnormal larvae. Kind of has warts, it looks really bad. And you can't really see the gut because it's so misshapen. And these abnormal larvae cannot successfully feed themselves. So they will most likely not develop into adulthood. And when we see more frequent events such as this, where we see higher ocean temperatures and also we'll most likely see it frequently and more intensely, we can assume that abnormal development will become more prevalent and also have negative consequences for future Lidocyanus pictus larvae populations. So we concluded that urchins that developed under more acidic conditions had higher tolerance to a thermal heat stress event compared to those that developed under less acidic conditions. And we also saw potential cross-tolerance exhibited in our urchin individuals. Cross-tolerance is basically just when an individual is exposed to one stressful event, they're better prepared to handle another different stressful event. And we also can conclude that abnormal development may soon become more prevalent as the increase in the frequency of learning interface. So in the future, I hope to analyze our morphometrics data and also explore other parameters such as gene expression, proteins, and lipids. And this can help us further understand the impact of environmental stressors on the physiological performances of Lidocyanus pictus. I wanted to thank my lab and also to all the CSTEP members who made this Gorman research experience possible. Thank you.